The first stop for carbon will be the Calvin cycle in photosynthesis. The carbon in the form of CO2 enters the Calvin cycle and bonds with 3 5-carbon molecules to form 6 3-carbon molecules. At this point, ATP is used and NADPH gives electrons to the 3 carbon molecules in order to form 6 G3P molecules, one of which comes out of the cycle. The other G3P molecule continues to the cycle to create 5 carbon molecules that continue bonding in the first place. When 2 G3P molecules combine, they form glucose, which is found in plants and allows humans to eat glucose. The plants are eaten by humans in a salad, and the human gets glucose in the plant to make energy through cellular respiration. The first step is glycolysis, where the glucose is broken into 2 pyruvate molecules. These pyruvate get changed into acetyl-CoA so that the Krebs cycle can use it. In the Krebs cycle, the 2-carbon acetyl-CoA bonds with 4-carbon molecule to make 6-carbon molecules. After that, CO2 leaves the cycle, and the 6-carbon molecules becomes 5-carbon molecules. Another CO2 is lost, and the molecule is lowered to 4 carbons. The other carbons go and restart the Krebs cycle, while the CO2 goes into the air and gets absorbed by plants through the stroma to allow the cycle to repeat. Thank you for watching our video about the carbon cycle.
So we know how the carbon cycle works, but how does CO2 leave the cell, and how does oxygen get into the cell? Now that is where transport across the cell membrane gets into action. If we zoom out from the cell membrane, we can see that proteins are not only useful in transport across the membrane, but also in DNA replication.
The cell cycle prepares the cell for mitosis. An important part of that is DNA replication, which occurs during S phase. To start off DNA replication, an enzyme called helicase comes between each pair of nitrogen bases and breaks apart the hydrogen bonds. Following helicase is an enzyme called primase. The purpose of this enzyme is to show polymerase where to start by laying down a primer so that the DNA polymerase can grab a hold. Then DNA polymerase latches onto the primer and builds DNA strands, reading 3 to 5, laying down 5 to 3. One of the strands does not follow the helicase as it unzips, and we call this the lagging strand. The DNA polymerase on the leading strand starts from the beginning of the primer and continuously follows the helicase as it unzips. On the other hand, the lagging strand does this process in the opposite direction in fragments, causing it to have to turn around and continue to repeat. Primer is also removed by DNA polymerase and replaces by the DNA strands. Then the last enzyme called ligase comes through, connecting any loose DNA fragments together. This forms 2 complete and identical DNA strands.
The cell cycle shows the whole life of a cell, from the creation through the life when the cell completes its function as it grows, and finally culminating in the process of mitosis. The whole cycle is preparing the cell for the process of mitosis, from the growing stages of G1 and G2 to the synthesis phase when the DNA is replicated. The first phase is G1, in which we see a new cell that has just been made in mitosis. This is the normal cell and interphase that is growing and doing its job in the body. Next we go to the S phase, also a part of the interphase. This phase prepares the cell for mitosis by replicating the chromosomes that then become sister chromatids. After that, the cell heads into the G2 phase to continue growing before entering prophase, the first phase of mitosis. In this phase, we have spindle fibers begin to grow from the centrioles, and the nuclear membrane begins to break apart. After the nuclear membrane dissolves, we move into the metaphase, where the sister chromatids line up in the middle of the cell and between the 2 centrioles. As metaphase continues, the spindle fibers grow longer and connect to the centromeres of the sister chromatids. After the spindle fibers attach, we move into anaphase, where the sister chromatids are pulled apart into the 2 different sections of the cell. The cell expands as the chromatids are pulled to either pole in anticipation of the
Now, while
division
cells
to
do
come.
everything in a body, nothing will
Anaphase
get done in
becomes
cells without
telophase,
proteins,
the final stage of
which are made
mitosis,
in the cells,
as the
like
chromosomes
this.
make their way to each pole of the cell. The large cell begins to pinch in the middle, creating a cleavage furrow as the cell begins to split. The nuclear membrane begins to reform and the spindle fibers dissolve. As the nuclear membrane forms, telophase and mitosis officially end, because mitosis is only the splitting of the nucleus, not the splitting of the whole cell. The cleavage furrow splits the cytoplasm in half and creates two new identical daughter cells through the process of cytokinesis. After that, the whole cell cycle repeats itself again and again for each new cell. Thanks for watching.
Protein synthesis by Susie and Kinsey. During protein synthesis, we start with a process called transcription. We start this process in the nucleus with a DNA strand. An enzyme called helicase unzips the DNA strand. RNA nucleotides make their way into the unzipped DNA strand and line up with their corresponding DNA nucleotide. The final product of this process is an inverse copy of the DNA, which is shown as transcripted RNA. To end transcription, the transcripted RNA
We've learned about
exits
many
the
functions
DNA
of proteins so
strand out of
far,
the nucleus.
but now let's explore the function of
Translation
proteins
takes
in
place
genetics
in the ribosome and has
using our
three main
MC1R
jobs, starting
gene.
with initiation. During initiation, the previous transcripted RNA, aka messenger RNA, is attached to a start codon, one by one. Transfer RNA attaches to the mRNA and standard groups of 3 RNA nucleotides, which is the start of the elongation period. The last group of tRNA to be added is a stop codon, whose job is to tell the tRNA when to stop attaching so that the amino acid chain stays the correct length. This begins the termination process. Attached to every tRNA group except for the stop codon are amino acids that bond together, forming an amino acid chain, which progressively folds into a protein.
This creates the final MC1R protein, which is the product of protein synthesis.
Genetics by Susie and Kinsey. In our study of genetics, we can look at the effects of certain genes on the fur color of a mouse species. In this experiment, we have a heterozygous dark-furred mouse and a homozygous
While genes are
light-furred
mostly
mouse.
consistent,
Each letter of the
sometimes a
genotype is
mutation can
representing
occur
a
in an
sequence of
organism
DNA
to allow
nucleotides
it
that make
to
up a
adapt better
specific
to
gene.
its
In this case,
environment.
our specific gene is fur color. We can use a Punnett square to predict the likelihood of the genotypes of offspring from our previous mice. Because the capital M is dominant and codes for dark fur, it overrides any light fur genotype that it is crossed with. In this case, the ratio of light to dark is 1:1, and the phenotype is 50-50. If we expanded the amount of offspring based on the percentages, we can show this prediction with a pedigree. In the case that there are 10 offspring, 5 will be heterozygous, while the other 5 are homozygous recessive, meaning 5 are dark and 5 are light. Thank you for watching our genetic stop motion by Susie and Kinsey. Evolution and Ecology, Biology Honors by Susie and Kinsey. In this representation, the light and dark beads represent our light and dark mice. Before the lava flow, white mice were the majority as they blended in with the environment better and avoided predator encounters more than dark mice. This dynamic changed when the lava flowed over the mice territory, causing the physical aspects of the environment to drastically change over time. The habitat went from light, with white mice being able to blend in, to dark as time went on and the lava hardened, switching to the black mice's advantage to blend in, surviving predators. Why do these differences exist? The dark mutation already existed, but slowly showed up less and less as it was not favorable in the lighter habitat. As is shown, the predator is more likely to eat more obvious mouse, which in this environment is the dark one. This led to the relative obscurity of the dark gene. Over time, this percentage of white mice significantly decreased due to the environment making it stand out more. This graph shows the trends of each population before and after the lava flow for each mice. Thank you for watching our Biology Honors Stop Motion Final.